All right, so as Warren said, my name is Ed Zaworski. I work with the Plant and Insect Diagnostic Clinic, where we process disease samples, not just for corn and soybeans, but a little bit of everything throughout the state of Iowa and neighboring states. And we're going to talk about scouting for field crop diseases. So I'm going to kind of break it down into two parts and cover what I can in a half an hour. I'm sure you've been hearing this all day. The half an hour is a short amount of time. I can't really talk about every field crop disease today, but I will cover a few, and mostly just, what goes into scouting for field crops. So the first slide here, I always like putting up this collage to show that field crop diversity in the Midwest is pretty diverse. You've got a lot of different diseases that affect at different stages, different plants, obviously. So it's quite diverse, and it can be quite challenging to identify everything out there while scouting. So every plant disease talk would be remiss if we didn't start off with what I call the disease triangle. There's three sides to a triangle, and there's three things that need to happen for you to have plant disease. First and foremost, on the left-hand side here, we have the pathogen, the disease-causing agent, the biological agent, whatever you want to call it. It might be fungi, bacteria, nematodes, viruses. So you need to have that, obviously, to have a disease. The other thing that seems pretty obvious is that you need to have a host plant, but not just a host plant, a susceptible host plant, one that can contract the disease. And what I mean by that is that not every plant is susceptible. Some plants, when perfectly healthy, probably won't contract a disease, but it may be if there's stress or something else going on with the plant, they are susceptible to contracting a disease. And then the third side, and probably the least obvious, is that you need to have a favorable environment as well. So fungi tend to like a humid environment, for example. So the weather dictates this a lot, and where you are climate-wise dictates this a lot. But you need to have all three of those things in order to have disease. So, like I said, the first part we're going to be talking about scouting in general. So I want to talk about what kind of background information you can gather before you even get to a field. And this could be generally, this could be area-specific, field-specific even. But number one seems very obvious. Of course, you want to know if you're dealing with corn or soybeans. But this is the number one point for me, because most diseases are specific to either corn or soybeans. They've evolved with corn or soybeans. That's not 100% true, but most of it is. So you can narrow down quite a bit by just identifying which one am I dealing with. Then knowing for a field or area, previous crops, adjacent crops, or non-crop areas. These are areas where we can build up inoculum, which would be spores or other things that are going to be disease-causing agents from year to year. So if your previous crop was the same as this year's crop, for instance, you've grown corn on corn, you have a higher chance of some diseases infecting that might have had a population last year and they've built up over the season. So what hybrid or variety you have. Some hybrids and varieties have resistance to pathogens, or they've been selected for resistance against pathogens. Recent weather, as I said, as going back to our disease triangle, we have to have a favorable environment to cause disease, so the recent weather is very important. The time of year, I'm sure you've heard this in several other talks already about weeds and insects. The time of year is very important as to what diseases are going to show up, both because of the weather, the climate, and because of the growth stages of the plants. Chemicals used on or near the crop are very important. Some chemicals can cause symptoms on plants that resemble diseases. So knowing the history of what kind of chemicals have gone on is pretty important. Planting depth and seedbed conditions. Again, some of these things, if done incorrectly, can resemble diseases, can cause stunting possibly. Soils and topography, again, another factor that is going to tell you. I mean, if your soils are well drained versus maybe like a heavy clay, the heavy clay is probably going to favor a seedling disease, for an example. So Current soil tests, so knowing your fertility, different nutrient deficiencies can manifest and cause symptoms very similar to some diseases. As an example, soybean sudden death syndrome can look quite a bit like several nutrient deficiencies in soybeans. And then your soil moisture and compaction, again, is your soil moisture high? Is the soil and is the soil compacted? I mean, compaction can cause, again, a lot of symptoms that are similar to young seedling diseases. So, all right. I like to show this slide. This is actually a slide showing a timeline of all the different growth stages in corn and different diseases that occur throughout these growth stages. So as you can see, early on from, VE to V6, we've got seedling diseases, obviously, right? That's the only time we have seedling plants. But then as the season goes on, different diseases, different foliar diseases, for example, usually favor different climates. So that's why we tend to see like a common rust showing up earlier than a northern corn leaf blight. And then later on in the season, we see an anthracnose top dieback or stalk rots and ear rots towards harvest. So, and I'd like to note, sorry, I probably should have mentioned this earlier. Warren has told me that he shared some links with everyone to our corn and soybean disease guides. And those are very helpful books. This is like a shameless plug for those. Those are very helpful books. Stuff like this slide, they will have kind of the probable growth stages that each disease will appear at. So highly recommend those books. So now that we've kind of gathered background information, we're going to think about what we need to do when we're scouting. And so I like to break it down into three main parts. And number one would be to look at the big picture or the field level, look at the little picture or the plant level, then collecting and recording information once you've had a good look around is also very important, depending on what you're going to do with the information you've scouted. So we're going to start with the big picture, and obviously, you've seen this again. This is going to be a rehash. I don't have a picture of the zigzag pattern, but examine the entire field, and I don't mean look at every single plant, and that's why a lot of my colleagues before me have laid out, hey, you know what, you want to scout in a pattern through the field, in an unbiased pattern to see what you can see. And so looking at the whole field, I like to ask myself, is there a pattern? There's a lot of different patterns that can occur. I'll show, my next slide has a couple of examples, but is there any kind of pattern to the symptoms that you're seeing in the field? And then number three, so are some of the things that we often associate diseases or other symptoms with would be fence lines, field edges, low areas, and exposed slopes. And then another thing to ask yourself while you're out there would be, are the symptoms that you're seeing in the field associated with any field activities? Sometimes it's pretty obvious that, if symptoms are following rows or if they're, following a spray pattern, sometimes there's very obvious symptoms that, or obvious patterns that are at the, that were probably associated with some planting activity. So I've just got a couple of patterns here to kind of lay out what you might see. And on the left, we have kind of a random pattern versus the right where we have an aggregate. Now, I don't like to live in a world of absolutes with diseases where one pattern or another means that there's disease or not disease, because every year somebody comes in with something and inevitably proves me wrong or something is amiss. So. But on the left-hand side, if you have a random pattern like this that encompasses the whole field, we're typically not talking about disease. We're probably talking about something else, something environmental, something else. Whereas with an aggregate pattern on the right, typically this is how a disease manifests. Typically we start in a small spot and it begins to radiate outward over time. And that's another way that you can kind of look at diseases versus non-pathogen symptoms, is that if they don't change over time, it's probably not a disease. Okay, so moving on to the little picture. So we're checking individual plants for symptoms and signs. So the first thing I like to do once I've identified an area where I think there's a disease is to compare a damaged plant, something in that area, to a healthy plant. I like to get a baseline when I'm out there. So every field looks a little bit different. Different hybrids and varieties look a little bit different. So I always like to grab a healthy plant and hold it up next to a damaged plant so I can get that nice baseline and see how bad it might be, you know. So the next thing would be to check the entire plant, which sounds obvious, but I used to crop scout myself, and when it's, you know, 95 degrees out there in July and you're looking for foliar diseases in a soybean field and the plants are lodged and you're having to kick through them, you're all beat up. Sometimes you you start to lack the willpower to look down in the lower canopy, but really you should check the entire plant. And then lastly, I like to bring a few tools with me, and nothing crazy, but if you've got one, bring a hand lens. You know, a lot of folks these days have microscopes or some kind of magnification device for their phone even, so you can look at stuff a little bit closer than you would be with the naked eye. A pocket knife for splitting stalks and stems, primarily in the late season, but something that's pretty useful to have in general. And then a shovel or just even a garden trowel would be useful. And the reason I say that is when people collect samples for us in the clinic, especially, and you know, if it hasn't rained recently, if it's relatively dry, when people go to sample something that they think might have a root problem, they tend to pull the plant out of the ground. And unfortunately, when that happens, what happens is that any rotted or necrotic tissue or the roots that you would want to see that would indicate a root rot disease are just going to fall off when you pull it out of the ground. So that shovel would go a long way to helping you look at that. I've just got some images here, you know, showing you upper canopy on the top left, inside the stems, outside the stems, and the roots. Just examine as much as you can if you're seeing something strange out there. And then the last part of the scouting bit here would be to collect and record information. And this could be tedious at times, like I said, especially if it's hot, but depending on what you're going to use this information for, if you're going to submit a sample to the lab, having as much information about the site and the situation is really important. So obviously what diseases are present, how severe are the diseases, and then what other factors could there be? I mean, we talked about knowing the chemicals and stuff. I mean, you don't always know that ahead of time, so maybe you're finding some of this stuff out as you scout, or if you're walking with a grower or a farmer, for example. So I'm going to talk about a few terms here in the next few slides. So when we're talking about assessment, there's two terms that you're going to hear. One is the disease incidence, which is, if you look at the picture on the left, oops. If you look at the picture on the left, the incidence would be the percentage of plants that are diseased. So if you look at that field, the percentage of those plants that are, you know, starting to defoliate, that's your disease incidence. Whereas severity on the right is showing on a per plant basis or a group of plants, how severe, what percentage of that disease or tissue is diseased. I've got an example in the next slide. So this is a disease rating for anthracnose stalk rot. So it's a 0 to 5 rating, where 0 is completely healthy, 4 and 5 have completely shredded pits, whereas 5 is lodged and 4 is not. This is just one example. There's disease severity ratings for a lot of different diseases. Some don't have them. Some people just use a percentage, kind of like Erin had in her presentation about percent damage from insects. So it varies across the board, but that's measuring severity. So this slide, we're going to have our first poll question, mainly to keep you awake for the last presentation of the day. And I'm going to ask you the unfair question of, are these two pictures showing the same disease? It's unfair because I haven't explained any of this, so, but I'm curious what you think. Rebecca, if we want to give them about 25 seconds, I think that's plenty. All right. Let's see, I can't see answers. So I'm just going to assume that you all, oh, there we go. So looks like about 38% of you, or of the ones that answered, got it correct, that they are not the same disease. It's okay if you got it wrong. I was intentionally trying to trick you and make you think here. So the disease on the left is actually bacterial blight in soybeans, and the disease on the right is Septoria brown spot. The one on the left is a bacterial disease, as it says in the name, and the one on the right is a fungal disease. And the reason I'm bringing this up is because I want to talk to you guys about symptoms versus signs, and basically what to do if you're stumped in the field. Disease identification in the field while scouting is not always easy, and it's not always possible. So I want to talk to you about symptoms and signs right now, and basically, if you don't know what it is, how can you find out what it is? So a symptom would be an adverse reaction to a disease, an environmental condition. You know, all of these spots on the leaves are a symptom. And in this case, the symptoms for these two diseases look almost identical. But if we were to go under a microscope and we were to look, we were to cut one of these lesions, for bacterial blight, we would see bacterial streaming if we put it in a little bit of water. We could even take it a step further. We could culture out the bacteria here. For the other one, Septoria brown spot, if you have your hand lens or other magnification device, you could kind of look a little bit closer at some of these lesions and see these small black pycnidia, which are spore-producing structures of the fungus. And the reason I bring this up, and I used these two as an example, would be, you know, if you're scouting for someone and you incorrectly identify bacterial blight as Septoria brown spot, or rather vice versa, Septoria brown spot as bacterial pustule, and the grower were to spray a fungicide, it would be completely ineffective on the bacterial disease. So I'm not trying to scare people off from making identifications in the field, but I am saying if you're not sure, try to get help. So I've got another poll coming up here in the next slide. Now, to test your knowledge, in this first picture that I've got coming up here, are we looking at a symptom or a sign? Again, the symptom would be the adverse reaction the plant makes, and the sign would be the actual spores, bacteria, etc. All right, it's about 50-50. So right here, we are looking at a symptom, and the reason is because we cannot see any signs of a fungus, bacteria, nematode, virus. We can't see any of those particles that would right now identify this as a disease. But if we were to take this sample, bring it in the lab, look at it under a microscope, we would find that within these black spots, that we would find our sporangia here. It's kind of odd not having faces looking back at you and people to ask questions, but I'm going to keep going. All right. One more thing here. The picture that I have here looks very similar to a disease, but it's actually a disease mimic. And what can happen with both corn and soybeans would be you have something that looks like a disease symptom, looks very much like a disease symptom. But most of the time when these symptoms appear, they're on 100% of a particular hybrid or variety. And then you might bring it into a lab, you might have it tested, and there are no signs. There's no fungi, there's no bacteria, there's no virus particles. And in this case, this is what we call a disease mimic. And it's not a disease, it's part of the genetics of the plant. And again, why am I going through all of this? Well, I'm trying to get you guys to reach out for help if you're unsure. So you have another thing here before we get into particular diseases, and that is what else is there besides a disease that could cause symptoms, adverse symptoms to a plant? And that would be disorders which are abiotic or non-biological. So they're not caused by a pathogen, they're caused by the environment or other things such as chemicals, compaction, nutritional deficiency, or as in that last example, genetics. So my last shameless plug, I plugged the books a little bit earlier, and now I'm going to plug the clinic where I work. So if you are unsure of what you're seeing, if you're not sure if you have a disease, or if you're not sure what disease you might be looking at, there are resources. The Plant and Insect Diagnostic Clinic at Iowa State is just one of the many resources. I don't know why that happened. You can also reach out to your regional field agronomist. There's lots of resources for folks out there if they need help. So feel free to contact the clinic. Feel free to contact your agronomist if you're not sure what you are looking at. Okay, with that, I'm going to move on from the scouting portion, and I'm going to talk about particular diseases. Again, I can't cover them all. Those books that I mentioned earlier are great. They have either a page or two-page profile of each disease and lookalikes and what growth stages they appear. So I'm just going to go over a few of the most common ones here because time is limiting. So first off, I'm going to start off with seedling diseases. For both corn and soybeans, there's 4 heavy hitters, and 2 of them are oomycetes, which are fungal-like organisms. 2 of them are fungi. So we have Pythium, Phytophthora, Rhizoctonia, and Fusarium. And these pictures here are illustrating kind of picturesque, picture-perfect examples of what seedling diseases look like on soybeans. It's not always this easy. In fact, most of the time it takes culturing and definitely looking at root tissue under a microscope to actually see which one of these you might be dealing with. But in general, you're going to be looking at stunting or discoloration of the roots or crown or cotyledon. So just got some take-home points for you about seedling diseases. Like I said, they're challenging, but you're going to look for discolored or mushy or squishy tissue compared to the firm white tissue of healthy root systems. And always make sure to note other variables. I've been harping on this for the most part through the whole presentation, and yeah, there's other things that can cause plants to stunt and not look right in the spring, and so make note of anything else that might be going on. So again, we're kind of rushing through, but like I said, I can't cover it all. So mid-season diseases would be next, and this is going to be mostly foliar diseases. And I think this time we're going to use corn as an example. So one of the most frequent things that I get in the clinic for confirmation, because they're kind of hard to differentiate, would be Goss's wilt versus northern corn leaf blight. On the left-hand side, you have Goss's wilt, which is a bacterial disease. On the right-hand side, you have northern corn leaf blight, which is a fungal disease. And again, going back to that earlier example, you can see how they can look similar. And if you were to misidentify one of them and apply a fungicide, it could be no good. So how do you determine which is which really quickly? I have a quick way to do so. Both of the pictures on the right-hand side are now Goss's wilt. And what are the differences between the two? So the Goss's wilt is caused by a bacteria, and it's going to cause water-soaked lesions. And what does that mean? It just means that the tissue becomes kind of wavy and it swells up a little bit, looks strange. And then it's going to cause what is a very scientific term, freckles. So all of these dark structures below that necrotic tissue up here are what I would call freckling caused by Goss's wilt. And this is the determining factor for me if I have Goss's versus northern corn leaf blight, is if you hold this leaf up to the light, then you're going to see what you see in the bottom picture here. These dark freckles will become translucent. Versus, northern corn leaf blight, I do want to point out that it can, Goss's wilt can cause these cigar-shaped lesions like northern corn leaf blight, but it will not have freckles. In some cases, people have sent stuff into me where they're sure there are freckles, and what's actually happening is that there's dark spots in these necrotic cigar-shaped lesions. They're actually just something that I refer to as junk fungi or saprophytic fungi that will colonize any dead tissue, and they will not turn translucent when held up to the light. So, late season diseases, I'm going to switch back to soybeans for this one. So 2 very common diseases in Iowa and the Midwest would be soybean sudden death syndrome and brown stem rot, SDS and BSR for short. And the trouble with these two and distinguishing between them is they make the same foliar symptoms. So this chlorotic or necrotic intervenal chlorosis, like this picture is showing, it would be the same for brown stem rot and SDS. So how do you tell them apart? Well, it's pretty simple, in fact. So if you look at these two pictures, I bet you all can guess which one of these is brown stem rot and which one of these is SDS. So brown stem rot, we traditionally have a brown pith. So that's the name, brown stem rot. Again, this is why you want to carry around that pocket knife when you're out scouting. Versus SDS, which is not going to have a brown pith and might have some pinkish brown discoloration around the pith. But again, both of them are going to have that same foliar symptom. A few more things that I want to touch base on really quickly. I haven't talked anything about nematodes. Nematodes are another category of pathogens on these crops in Iowa. The most common and most damaging would be the soybean cyst nematode, which typically has the highest yield impact in Iowa. And for the most part, this is a hard one to scout for because you can see the picture in the upper right where there's stunted yellowing plants. For the most part, you'll never see this. For the most part, it's asymptomatic like this. Most of the soybeans that are, if not all of the soybeans that are planted in Iowa have resistance to soybean cyst nematodes, so they almost never show drastic symptoms. So it's kind of one that if you're scouting a field where, you know, something's not quite right, the yields aren't quite right year after year, you might want to have the grower sample for a nematode, and that's done through soil sampling in a lab. And then I think the last thing here that I have before a few notable diseases would be viruses. I'm not going to talk too much about viruses. It's not something I typically expect myself or others to be able to identify or diagnose in the field. For the most part, these are going to require DNA-based or serological-based assays to identify them. You can definitely tell what looks virus-like, though, and so something like one of these pictures, if you do see it, you might want to get a hold of a lab if it's a big problem, or if you just want to identify what it is. So we're coming up on my last poll here. Again, it's a pretty unfair question, but it pertains to tar spot. I'm sure at this point, many of you have heard of tar spot. It came onto the scene in 2016, and my question to you would be, how many Iowa counties has tar spot been found in as of 2020? Again, this is an unfair question. I apologize, but I'm just trying to keep you, make sure you're awake here. It's almost over. All right. All right, so the general consensus is that it's been about 34 counties. Well, thanks to the Integrated Pest Management Group and other teams on campus here last year, we were able to scout a huge, most of Iowa, in fact, and we were actually able to find tar spot in 75 counties. So it is definitely out there. It was at low levels in many of those counties, but I'm pointing it out because we do want to hear from people when they do find this disease. There's been several notable situations where this disease in high severity can cause pretty bad yield problems. So we definitely want to hear about it if you find it, and we definitely want to hear about it if you're in a county that has not seen it yet. And then I've got one more here. This is another disease that came onto the scene relatively recently. We've definitely not seen it in as high of incidence as tar spot, and we definitely have not seen it cause any problems yet, but it's another one that if you do see, I'd like to hear from you. And it can look a lot like another disease, gray leaf spot, which I didn't cover in this, but again, very much like Goss's wilt, the other bacterial disease on corn, the other major bacterial disease on corn. When you hold it up to the light, the lesions are going to look quite different, as you can see in this picture in the lower middle portion. So with that, I gotta be getting close to time, so I'll summarize here. There's a lot of things to consider, gather information on your hybrid variety, planting and management practices. I'm sure some of my other colleagues have mentioned watching the news can sometimes give you a heads up, particularly if there's something new. Know your weather conditions, your big picture, your little picture. Use references. I really do recommend those books. I'm not just trying to get you to buy them. They are really good resources. So, yeah, and ask questions, reach out to folks. I've got contact information for myself, our corn pathologist at Iowa State, Allison Robertson, and our soybean pathologist, Darren Mueller. The information for contacting the clinic here is on the top of the page. If you're unsure, our motto is always test, don't guess. So with that, I'll take any questions that have built up. Everybody's just ready to get out of here.
I do have 2 questions for you, Ed.
Okay.
The first one is, when you take a diseased plant and you isolate an organism in the lab, sometimes you might get multiple pathogens that show up. How do you know which one is the pathogens that's actually causing the disease, or how do you confirm what is the specific pathogen?
Absolutely. So for the most part, it depends on what kind of disease we're talking about. Generally speaking, if we're talking about a root rot pathogen, I don't think I've ever seen, like I said, if you can't live in a world of absolutes with diseases, but I rarely see co-infecting root rot diseases. But then beyond that, if we're talking about a foliar disease and we're trying to do isolations to determine what it is, you're gonna rely a little bit on the symptoms that you're seeing as well as what you're isolating. So that's the thing. With any diagnostic techniques, it's hard to rely 100% on any one. So for the most part, we're looking at the symptoms, we're looking at what signs we find under the microscope as well as what we've isolated. So yeah, you'll typically be able to tell. Not even with the seedling diseases, which are traditionally very hard to identify, there might be some symptoms that you can differentiate between the two, or between 4.
The other question that we had, with tar spot being a new disease, what are some tips for diagnosing or identifying tar spot in a field if you don't know what you're looking for?
That's a really good question. So again, face-to-face, I have laminated examples of tar spot, but what I would say is that tar spot is very unique. Those black stroma that are popping up on the top of the leaf, they're very hard. So I think that the key that I would say is, if you think you've got it, take your fingernail and scrape those black lesions like that. And if you can't scrape them off, then it's likely you have tar spot. But having scouted it last year, there's a few things that can trick you, but if you can't scrape it off with your thumbnail, you're probably talking about tar spot.
And then the other question related to tar spot is, do fungicides help?
Right. So this is part of the reason why I mentioned tar spot, because research is still undergoing, and people are trying to figure out, what I do know and what I can tell you is that preliminary research shows that fungicides do have an effect. I can't really speak to which fungicides are best at this point because it's still being studied, but it's been perceived that fungicide does make a difference with regards to tar spot. But it also depends on when the disease appears, how severe it is, when the spray occurs. So.